Hello, myself Varsha Burman, and I'm going to deal with the topic that is bioinorganic chemistry. So, as we all know that bioinorganic chemistry is the most recently explored field of the inorganic chemistry, and the main task of the bioinorganic chemistry is to identify the inorganic compound involved in the various biological process, to study these compounds and to control and optimize the system. And bioinorganic chemistry deals not only with the elements and their compounds that are present in living organisms, but it also deals with those organisms which are present in the atmosphere. So, among the tasks of inorganic biochemistry is the investigation of the structure and biological role of the inorganic compound. So, from the chemical point of view, uh, the bio in living beings there are two type of constituent. In living beings there are two type of constituent which are present. The first one is organic constituent and second one is inorganic constituent. So in on the basis of organic and inorganic constituent we have to study the rest of the chemistry. So let's go to the next page. So first of all, let's go to the introduction. So, bioinorganic chemistry is the field that examines the role of matter in biology. As we all know that the various inorganic elements which are biologically important in our human body. So, bioinorganic chemistry is the field which examines the actual role of these metal ions in our body. And bioinorganic chemistry includes the study of both natural phenomena such as behavior of metalloproteins as well as artificially introduced metals, including those that are non-essential in medicines and toxicology. Many biological processes such as respiration depend upon the molecule that fall within the realm of inorganic chemistry. The discipline also includes the study of inorganic models of mimics that imitate the behavior of metalloproteins. So, in terms of bioinorganic chemistry, we have to study various type of metalloproteins which are present in our body, such as hemoglobin and myoglobin. And in hemoglobin and myoglobin, iron atoms are present as inorganic elements. So, the main role of bioinorganic chemistry is to deal with this type of elements which are biologically important in our body. So, let's go to the next page in order to study the rest of the things. So the bioinorganic inorganic chemistry is about the structure, function and mechanism and dynamics of the biologically relevant metal complexes and metal containing proteins. Okay? And metal containing protein. Investigators in the area try to understand what the functions of biological relevant metal site, how it achieves its function and what factors dictates its function through studying either small metal complexes or metalloproteins. So, biologically, or in terms of biological manipulation, theoretical calculation, and advanced characterization techniques, including single molecule spectroscopy, ultrafast spectroscopy, and synchrotron based X ray spectroscopy and diffraction studies, are used in order to study the role of these metalloproteins in the biological system. So, on the basis of bioinorganic chemistry and on the basis of bioinorganic inorganic elements which are present in our human body, we are going to study hemoglobin. Where heme means iron and globin means protein. So, heme protein. So, hemoglobin is an iron protein which is present in the human body. So, hemoglobin is actually abbreviated as HP or HGB. It is an iron containing oxygen transport metalloprotein in the red blood cells, and it is also known as erythrocytes and found in various vertebrates and also in the tissues of some invertebrates. And the main role of hemoglobin is to carry oxygen from the lungs to the different body to the different tissues of the body. Okay, there is there in the tissues it releases the oxygen to permit aerobic respiration to provide energy to power the functions of the organisms in the process called as metabolism. A healthy individual has 12 to 20 grams of hemoglobin in every 100 ml of blood, as we all know that. So hemoglobin is involved in the transport of other gases. It carries some of the body respiratory carbon dioxide, that is about 20 to 25% of the total, and in which CO2 is bound as heme protein. This molecule also carries the important regulatory molecule nitric oxide bound to the globin protein thiol group, releasing at the same time as oxygen. Other important point of hemoglobin is that hemoglobin is involved in the transport of other gases, and that other gases includes and that other gases carry some of body respiratory carbon dioxide, about 20-25%, as we already read in the previous slide. It is actually repeated, sorry for that. So now we come to the structure of hemoglobin. What is the structure of hemoglobin is all about? So hemoglobin is a quaternary structure, and it is a characteristics of multi-subunit globular proteins, and it is found in various vertebrates. The hemoglobin molecule is an assembly of four globular protein subunits, and its subunit is composed of a protein chain tightly associated with a non-protein prosthetic group. So the non-proteinous part is called as the prosthetic group in hemoglobin. And a heme group consists of an ion held in the heterocyclic ring known as porphyrin. So the porphyrin is a tetrapyrrole structure which is made up of four imidazole ring, and this porphyrin ring consists of four pyrrole molecule. As I already tell that the porphyrin complex is made up of, it is a ring structure which is made up of four pyrrole molecule of imidazole ring. So, porphyrin ring consists of four pyrrole molecule cyclically linked together by methyne bridges with an iron bound in the center. The iron, which is the site of oxygen binding, coordinates with the four nitrogen atom in the center of the ring, which and all they are lie in one plane. The iron is bound strongly covalently to the globular protein via the nitrogen atoms of the imidazole ring of histidine residue, which is already also known as proximal histidine, below the porphyrin ring. A sixth position of the hemoglobin is occupied by the oxygen by the coordinate covalent bond, completing the octahedral group of the six ligand. The reversible bonding with oxygen is why hemoglobin is so useful for transporting around the body. And the iron which is present in the hemoglobin is either in ferrous form, means iron can be present either in Fe2 plus form or Fe3 plus form, but it is impossible for the but iron in Fe3 plus form cannot bind oxygen. So in order to bind oxygen, if metmoglobin reductase and in no, metmoglobin reductase will be able to reactivate itself by reducing the iron center, and by reducing the iron center, the Fe2 plus is now capable of binding the oxygen. So the iron in Fe2 plus oxidation state will bind the oxygen. Carry on to the further page. In adult human, the most common hemoglobin type is a tetramer, which contains four subunit proteins called hemoglobin, consisting of two alpha and two beta subunits non-covalently bound, each made up of 141 to 146 amino acid residues respectively. This is denoted as alpha 2 beta 2. The subunits are structurally similar and about the same size. Each subunit has a molecular weight of about 16,000 daltons. So 4 subunit is equal to 64 daltons. So this is all about hemoglobin structure, which is present as a metalloprotein in the human body. So next page is all about the structure. So here's the structure of hemoglobin. In the center, iron is present and it is tetrahedrally surrounded by the four nitrogen atom, which comes from the pyrrole ring of the imidazole molecule. And the fifth position may be coordinated with water, or the sixth position is coordinated with the no sixth position comes from the nitrogen atom of imidazole ring, and the sixth position is occupied by the oxygen atom. So this is all about hemoglobin. And now, thank you.